Technical issues. I hope I don't bother you with my technical issues if potentially something happens. Have you ever wondered how our brain is structured or wired? Our brain is composed of billions of neurons and fibers. And these neurons and fibers are responsible for transmitting electrical signals among each other. And neurons and fibers constitute a sort of cable salad in nanometric scale. Can you imagine tracing a single wire in this cable salad? According to legend, Alexander the Great had a similar problem, which is known as Gordian knot. And his solution to that problem was just cutting the wire salad or the cable knot by his sword. Of course, neuroscientists work more elegantly. What they do is they take a small piece of brain tissue, and they cut this tissue into very thin slices. And from each slice, they take an electron microscopic image, and they end up with a 3D image stack, a volume of images. After that, they need to annotate the corresponding cross-sections in each slice by slice, and link these tabular objects. This operation is mainly done by semi-automated manual input and semi-automated annotation systems. And the domain is called connectomics, and it is sort of a brain mapping process. In the neuroscience literature, there are certain applications, in the neuroscience domain, there are certain applications which are based on manual annotation. Here we see EyeWire, and on the right-hand side, you can follow maybe the manual input with the mouse cursor. As you notice, from time to time, the user has to pivot the system to let the automated system know where the traced fiber, the cross-section, is located. In this talk, I will present you not the manual annotation case, but the gaze input case. Our system is called iTrace, and I will also talk about our user experiment results. But before that, what is the shortcoming of manual annotation? In neuroscience domain, people have done a series of research, and Takamura et al. Has presented their results in Nature 2013. In this work, they have presented successful trace of 380 neurons, and it took them 15,000 person hours. That means 20 people, around 20 people, a lab full of people, were working half a year long only for annotation task. And this was only actually mapping not a human brain, actually it was a fruit fly. And from human-computer interaction literature, actually we know that the manual annotation task can be described as a function of fiber size and the distance of the fiber to the target size and the location of the target to the mouse pointer. And several researchers in the Chi domain and in HCI domain tells us that potentially gaze interaction, gaze input can be replaced, a manual input can be replaced by gaze interaction. So I just want to test whether you agree with this hypothesis. Can we replace manual input, manual interaction with gaze input? Here we have a test data, and I want you to just select a particular cross-section, something like this or this, and then just try to follow as the simulation, the animation progresses. I'm sorry. I need to, okay. Were you able to follow the cross sections with your eyes only? Maybe a few hands? Okay, thank you. We had this same intuition, and with this motivation, we have implemented our iTrace system. Our system makes use of several open source libraries and tools. On the visualization side, we made use of imagery and SIGCHI, and in the orchestration for experiment, we made use of the MIGI, which is an extension for the imagery. And in the interaction side, we had a low-budget, low-cost eyetracker, which is eyetracker. And for the interaction as a second modality, we made use of a gamepad controller. With the gamepad controller, the user was allowed to move and pan and zoom the visualization, and also start and stop the tracing trials. Here you see our system in action. We had to use a chin rest, as you might have noticed, because the accuracy of the eyetracker was not as high as we wanted to, but it was a low-budget solution, so for gamification purposes, it was potentially actually ideal. Our user experiment has investigated two main questions. We wanted to know how the tracing accuracy was affected in terms of tracing speed, represented with this symbol, and of course the size and morphology of the fibers from large, medium, and small categories. On the second side, second part, we were interested in how the repetitive trials of tracing same fiber was affecting the tracing accuracy. In this category, we were only using one particular tracing speed, which was 20 frames per second, but I will come to this later, the details later. Prior to our experiment, we have prepared ground truth data with manual annotation. This has been performed by an expert neuroscientist at 5 frames per second approximately. And we have measured the accuracy by means of root mean square error and the comparison of the participant trials with the ground truth data, of course. Our fibers were then categorized, the selected fibers were then categorized in 3 different morphologies, denoted by large, medium, and small. And these are measured in pixels, of course. In our experiment, 20 graduate and undergraduate students have participated. We had to discard one participant, the results of one participant, because it was definitely, obviously, an outlier. The details are in the paper. And the experiment, which I'm presenting here, has composed of 3 main stages. In the first stage, in the training phase, the participants have given their consent first, have asked to give their consent for this experiment, and they have been trained for different fiber morphologies in these categories, and then they were trained for using the gamepad for panning and zooming and starting and stopping the tracing. And also they have been informed about the eye-tracking interaction, and a 9-point eye-tracking calibration has been performed. In the second stage, which was actually the main part of this experiment, we have tried to answer the first question, the effect of tracing speed and fiber morphology on the tracing accuracy. And in the last stage, we were interested in the effect of repetition on tracing accuracy. So in the main experiment part, the participants were asked to perform 48 trials. This is actually a basic math. It was the experiment in the experiment only 12 different fibers, unique fibers, were included from 3 different categories, and we had 10, 20, 30, and 40 frames per second as speed levels. Of course, the list of trials were randomized to avoid learning effect. And at the end of each trial, we asked the participants to rate the level of difficulty of tracing that fiber from 1 to 5 on a Likert scale. 1 was very easy, and 5 was very difficult. For the third part of the experiment, in the repetition session, the participants were asked to do 8 order trials, so no randomization, of course. In the first part, they have been asked to trace the same small fiber for consecutive times, and in the second part, a medium fiber for consecutive times again. Our tracing speed was 20 frames per second, constant, and the participants have been asked again for rating the difficulty of tracing that, making this task. And our results looked like this. In the speed, size, accuracy experiment, for large fibers, we were not able to observe any significant effect of working with large fibers on the tracing accuracy. There was no significant effect. Here you see actually a sample data set. This is actually the boundaries of our fiber, which has to be traced. I'm sorry. And the dashed lines represent the average participant performance in 20 and 40 frames per second. As you might have noticed, the participant results are remaining within the boundaries of the large fibers, representative large fibers. And in terms of medium and small fibers, we have noticed, we have observed significant difference, as you might have noticed that the boundaries are actually exceeding the representative fiber boundaries. That means we had actually less accurate results. So these fibers were more difficult to trace in terms of accuracy. The repetition tasks, the repetition experiment has shown that we were not able to observe any significant differences in terms of tracing accuracy. In terms of task difficulty, we had some interesting results. As you see on the left-hand side, the difficulty ratings were proportional to the tracing speed. So the more speed we had, the more difficult the tasks were perceived. In terms of fiber size, the small fibers were significantly more complex to trace. And on the repetition side, on the right-hand side, we had only observed one certain case, which was the tracing small fibers. There was a significant difference in terms of difficulty ratings between the first and fourth trial. That means for small trials, small fibers, the repetition might help to trace and learning tracing actually at the end. Yeah. In conclusion, I tried to summarize the take-home messages. So we tried to implement proof of concept type of test bed. And our user study results show that actually we had a certain accuracy. We had other results in our paper which are also significant to note. I mean, I suggest you to read that. But in summary, mouse was more accurate than gaze, roughly speaking. But we could achieve more speed in terms of by using gaze interaction. As we use the low-budget eye tracker, we are planning to use a better eye tracker to continue our research, and we see potential of using our system for supporting proofreading tasks for current semi-automated image annotation pipelines. Thank you.
Oleg Komogortsov, Texas State University. Very nice work. If I may, suggestion. So as you correctly mentioned, you are using eyetracker, so it has some accuracy characteristics. So it's very interesting to have maybe another objective point if you're going to use a more accurate eye tracker in terms of how much faster you're going to be. Maybe you're going to be even faster and more accurate, right? Because actually,
that's our goal.
iTribe has some very challenges in terms of accuracy.
We observed that also.
Yeah.
Thank you.
Thank you.
Hi, nice talk. Quick question. So your mouse-based segmentation was done by an expert, right?
That's right.
And the gazing was not done by experts, right? So
the gaze tracing was done by participants.
Okay, so maybe it would even be better with an expert, right?
Of course, but we wanted to test with novice users, and we wanted to see the potential of using this system for maybe potentially for gamification aspects.
Okay.
That's why we use like novice users also. But partially they were from the neuroscience domain, so they were not totally unfamiliar with what was going on.
Okay, cool. Thanks.
Thank you.
You're listening to your speaker, and I'll let